Why, hello there, and welcome to a new segment that I'm calling Get Nerdy With Me. Today I'm going to be talking a little bit about something that's been appearing in the news lately and generally referred to around the discussions pertaining to the new coronavirus vaccine. We're talking about monoclonal antibodies. For those who don't know, I am a graduate student here in Sweden studying experimental and medical bioscience. And I'm going to be trying to fill in some of the holes left behind in popular media and news as they tend to gloss over some of the more pertinent details of their scientific explanations. Let's get started. So, what are monoclonal antibodies? You usually see monoclonal antibodies anytime you're talking about vaccines or medical treatments, but sometimes also different tests. For our purposes, in order to understand what a monoclonal antibody is, we need to start with this word. This is an antibody. It has a couple of different regions that are important to how it works. They are made by your immune system to help fight off disease, infections, and generally keep you alive and healthy. Up at the top, we have two Fab regions. And no, that does not stand for fabulous. These are what create the epitope, or the type of antibody that this responds to. Think of it like a lock. Each lock requires a different shaped key, and in order for this antibody to get to work, it has to latch on to that specific key. The bottom part is called the Fc region, with C standing for constant. This part doesn't change. Now, there's something very important that you need to understand before we can go any further. Antibodies react to antigens. Antigen stands for antibody generation. So if antibodies react to antigens, then what's an antigen? An antigen is anything. It could be anything that triggers antibody production. And I'm not trying to be facetious here. It could be a chemical, it could be a type of sugar or fat, but generally speaking, it's a protein. Not exactly like the fried eggs type of protein, but you get the picture. More like this kind of protein. Proteins are little squiggly masses of amino acids that are combined together. Our bodies use them to make fuel and muscle, and they're generally a useful thing to have in our systems. But proteins have specific little sequences on them. These sequences are what make the protein what it is, but they are also what our antibodies are reacting to. And they come in different types, different shapes, different keys. Starting to get the picture? Each one of these different nodules on our protein is going to create a different antibody. So, how do you make an antibody? Well, vegetarians, this is where you should look away. Unfortunately, antibodies can only be made by using a biological organism. Sometimes this is something like a bacteria or plasmid, but generally speaking, when we're talking about antibodies, we're talking about thumper here. You inject the animal. Now remember, antibodies are made after exposure to an antigen. So to start off with, we're going to inject Thumper here with our antigen. That's the protein with those specific sequences on it. Once that serum enters Thumper's bloodstream, Thumper here will start to create antibodies. But remember, our protein had a bunch of different types of sequences. So Thumper will actually make different antibodies for each sequence. Thumper will make multiple clones of antibodies, also known as polyclonal antibodies. And unfortunately, we can't just separate them out with our colorful test tubes like we see here. Once antibodies are made, they're kind of mixed together. In order to create separate antibodies, you need to start with separate antigens. You need to cut out the sequence that you're looking for. But that's easier said than done when each sequence could have multiple different sequences or multiple antigens. Even though we cut a segment and think we've isolated the antigen of choice, after we inject it into Thumper and Thumper makes the antibodies, as it turns out, we may still have multiple types of antibodies. So we cut again, and again, and again, and again, and as many times as we need to, until we have isolated the antigen that produces the antibody we are looking for. Because we can't actually see what's happening inside of Thumper, there's a little bit of trial and error here. But sooner or later, if you're lucky, you are able to successfully isolate one type of antibody, or a monoclonal antibody. Now, to be clear here, monoclonal antibodies aren't necessarily better. Sometimes polyclonal antibodies can actually give you a better result. It just depends on your purpose. But generally, monoclonal antibodies are what we're looking for to fight specific diseases. They're the magic tool in our healthcare arsenal that help protect us and keep us safe from disease, particularly from COVID-19. In general, monoclonal antibodies are incredibly difficult to produce, can be very expensive, and extremely temperamental. They're also the crucial bit of research that the new Pfizer vaccine is based on, as well as the Moderna vaccine and the research currently being conducted in China. Until we have a vaccine that is able to reach everyone, preventative measures are the best steps we can take. But with new breakthroughs in monoclonal antibodies, hopefully we'll be seeing these vaccines available to consumers soon. I hope this was helpful, and if you guys want to see more sciencey nerdy bits, definitely let me know. And until next time, do good.